Hello and welcome to your lecture on lipids. So lipids are molecules that contain many carbon-hydrogen bonds with little to no oxygen. Carbon-hydrogen bonds are nonpolar. They're nonpolar because both carbon and hydrogen have very similar electronegativities, and therefore they both share electrons equally in their covalent bond. Because lipids contain so many carbon-hydrogen bonds, generally lipids are nonpolar. Lipids, of course, don't mix in with water. You probably did an experiment in preschool where you tried to mix oil with water and it didn't work, and that's because the oil is a lipid which is not soluble in water. What makes the lipid not soluble in the water is the fact that water likes polar molecules or charged molecules, and lipids are nonpolar. There are three main groups or categories of lipids: triglycerides, which are also commonly called fats, phospholipids, and steroids. Triglycerides and phospholipids both share a common component to them. It's a structural component called a fatty acid. Steroids lack a fatty acid. What unifies all of them as a lipid is that they all contain these many carbon-hydrogen bonds, making the molecules generally not soluble in water. There is no single common monomer to a lipid. Carbohydrates had monosaccharides as a monomer. Proteins have amino acids as a monomer. Nucleic acids had nucleotides as a monomer, but there is no true monomer to a lipid. Fatty acids come close, but because the steroids lack a fatty acid, we can't truly say that fatty acids are the monomers of lipids. Nonetheless, fatty acids are important because they're a common component to two of the three groups of lipids. So fatty acids are just really long chains of carbons and hydrogens. Because of this, they're sometimes referred to as hydrocarbon chains. Normally I would draw fatty acids for you on the board, but because we're doing this lecture capture, I'm going to show you some pictures of the two different types of fatty acids that exist. They're called saturated and unsaturated fatty acids. Okay. So up top you can see what's known as a saturated fatty acid, okay, and down below here is an unsaturated fatty acid. You can see we have a long chain of carbons and hydrogens. Right? Over at this end, this is the acid part of the fatty acid. This hydrogen can leave, and of course anything that can donate hydrogens is considered an acid. The difference between these two is, in fact, the presence of a double bond here in this unsaturated fatty acid. Now, an unsaturated fatty acid can have more than one double bond. This is an example of something called a monounsaturated fat, but polyunsaturated fats do exist. The saturation is referring to saturation with hydrogen. This fatty acid up top here, the saturated fat, contains all the possible hydrogens that it can. It's completely saturated with hydrogen. This one is not completely saturated with hydrogen. We could technically fire hydrogens at this carbon-carbon double bond. That would break the carbon-carbon double bond, forming a single bond between these carbons, and then the carbons would then take on more hydrogens. You can see that they are structurally different. Notice the nice straight saturated fatty acid chain, whereas with the unsaturated fatty acid chain we have a bend. Those carbon-carbon double bonds produce bends. And because these things are structurally different, they are functionally different in our bodies. You're probably more familiar with the term saturated and unsaturated fats as we use them applied to foods. Animal fats such as butter tend to have a large amount of saturated fat in them, whereas plant fats such as olive oil tend to have a large amount of unsaturated fatty acids in them. And these things are so different that at room temperature, the saturated fatty acids are solid, whereas at room temperature the unsaturated fatty acids are liquid. So these are very different substances, and they do act different in our body. So this is just a picture of something called the triglyceride that we'll talk about in a minute, but you can see how there's 1, 2, 3 long chains of fatty acids here. This is also a triglyceride, but you'll notice that we have double bonds present. So these are unsaturated fatty acid unsaturated fatty acid chains over here, whereas these fatty acid chains are saturated. And notice how different the ultimate shape of the molecule is, depending upon whether or not it contains saturated or unsaturated fatty acids. So the first category of lipids that we're going to talk about are the triglycerides, or fats. The picture that I just showed you was an example of two different types of triglycerides. The tri in triglyceride is referring to the fact that they contain 3 fatty acid chains, and the 3 fatty acid chains are held by a glycerol molecule. So this is the glycerol molecule here, and then you have 1, 2, 3 fatty acid chains attached. Notice we have 1, 2 saturated fatty acid chains, and the third one is unsaturated. This is a great molecule for energy storage. We know that carbohydrates are used for energy in our bodies, but the triglycerides provide a lot more energy. The energy in carbohydrates and the energy in fats comes from carbon-carbon and carbon-hydrogen bonds. And so we have many more carbon-carbon, carbon-hydrogen bonds in triglyceride than we do in, say, glucose. Right? Glucose only has 6 carbons, right? So there aren't that many bonds, aren't as many bonds in glucose as there would be in a triglyceride. Triglycerides tend to be stored in fat tissue, adipose tissue. The cells there are called fat cells or adipose cells. Our next group of lipids are the phospholipids. They're very similar to a triglyceride. Basically, if you take a triglyceride, remove one of the fatty acid chains, and put a phosphate group in place of that fatty acid chain, you essentially have a phospholipid. So they still have a glycerol backbone, like a triglyceride. They have 2 fatty acid chains, but then they also have a phosphate group. Right? So this is an icon model of a phospholipid here. This is one that you're going to see very often throughout the semester. And here's our structural formula for a phospholipid. We have 1 and then 2 fatty acid chains. In red, you have your glycerol. And then here, in place of the 3rd fatty acid chain, you have a phosphate group with some other things attached. Notice the negative charge in the phosphate group. That's very important. That phosphate group is hydrophilic, and the reason why it's hydrophilic is because of the charge on the oxygen there. Remember, water likes things with charge or partial charge. So here we have a phosphate group that has a full negative charge. So the hydrogens on a water molecule will be attracted towards that negative oxygen in the phosphate group. The fatty acids are very much hydrophobic. So these 2 chains here of fatty acids are very hydrophobic. There's a large number of carbon-carbon, carbon-hydrogen bonds present, and those are non-polar covalent bonds, and they're hydrophobic. So if you look at this phospholipid, you'll see that we have a hydrophobic part and a hydrophilic part. Right? We call this upper area the head of the phospholipid, and it's said to be hydrophilic because of the phosphate group. And then these 2 fatty acid chains are said to be the tails of the phospholipid, and they are very hydrophobic. So the phospholipid is both hydrophilic and hydrophobic. We have a special name for that. It's called amphipathic. So phospholipids, because they have this interesting property of being both hydrophilic and hydrophobic, they do something very interesting when you put them in water. When mixed with an aqueous or water solution, phospholipids spontaneously form one of 2 things. They either form a phospholipid bilayer or something called a micelle. So a phospholipid bilayer suggests that you have 2 layers of phospholipids. Right? What happens here when you try to mix these phospholipids with water is that the hydrophilic, those water-loving regions with the phosphate groups on them, they remain exposed to the watery environment because the water is attracted to the phosphate group on the hydrophilic head. The hydrophobic regions of the phospholipids tend to group towards one another. So those fatty acid chains tend to group together, and that's because they don't like the water. They're trying to exclude the water from themselves. And these phospholipid bilayers form the basis of a cell membrane, and they form the basis of many of the organelles we find inside of our cells. What I'd like you to do is try to draw a phospholipid bilayer surrounded by water. Include 6 total phospholipids and indicate where in your picture is the water. So take a minute to try to draw a phospholipid bilayer. You can pause the slide here. So hopefully you drew something like this. This is a phospholipid bilayer. The blue area is the water, right? The circles are the heads, the hydrophilic regions of the phospholipid. They are facing the water because they contain those charged phosphate groups that like the water. Okay? So you can see on both sides the heads are facing the water environment, whereas the fatty acid tails are facing one another. They've excluded the water from themselves. Another thing that can form when you put fatty acids in water is this structure here called a micelle. It's basically like a sphere of phospholipids. All the hydrophilic heads are facing the outside watery environment, whereas all the hydrophobic tails are facing one another. Now, both of these do exist in our bodies, but we're going to talk a lot about phospholipid bilayers in the future. Micelles are important for things like digestion. Our last group of lipids are the steroids. Like the other lipids, they are lipid soluble, but they look pretty different from the other lipids. They don't have a fatty acid chain, so there's not a long chain of carbons and hydrogens. Here we have, the shortest chain of carbons and hydrogens, but not considered a fatty acid. A common component to steroids are these cyclic groups, right? So where all these carbons are forming these cyclic groups, that's very common to a steroid. Okay? Steroids include things like cholesterol, the hormone testosterone, and the hormone estrogen. So here we see cholesterol. Actually, many hormones like testosterone and estrogen are actually made from cholesterol.